generation of a different cohort. We recently set up our own BL2+ space, so you can see that the first one took a little bit longer, and they're getting progressively faster as we automate the process and kind of optimize aspects of it. So more specifically, just for kind of big picture perspective, the way that we generate our data is we, select genes for use in each cohort, and then our genetic perturbations platform here at the Broad generates virus plates for us using lentivirus. And separately, we derivatize the cell lines that we treat these treat the virus in. So these are 2 vector systems. Before we screen at scale, we do a number of optimization and pre-screening procedures, mostly to make sure that infection efficiency is where we want it to be. And then we do the actual screen itself, where for each virus plate, we treat it in each cell line, and then also set aside 2 plates to treat with puro and no puro to assess infection efficiency. And the screen itself is done in triplicate. That's what those 3 plates down at the bottom represent. And those are what we use to actually generate our signatures. So just to give you guys an example, I've included a sample plate layout. The main point of this is just to kind of represent the types of perturbagens in each plate. So very importantly, we have landmark genes distributed throughout the plate. Again, these are the directly measured ones. In this top left corner, we have CMAP controls, which are just platform controls. We don't actually use those for any data. The light orange represent our reference genes. So these are well-performing guides that we include in the same position on every plate, just to have a reference, both in terms of technical quality and to be able to assess things like batch effects, reproducibility across cohorts, and so on. Next, we have negative controls. These are guides that don't target any part of the human genome. And we have untreated wells as well, which are just derivatized cells that don't receive the second virus treatment. So once the data is actually made, we go through kind of our standard data processing pipeline. I won't go through all the details, although I'm happy to, again, discuss this in further depth with anyone who's interested. But basically, we normalize our data, we infer the expression of the genes we didn't directly measure, and then we calculate differential expression z-scores and aggregate the 3 replicates that we have of every plate into a single signature. So when I show you results later down the line, I'm going to primarily be talking about the level 5 data, which are differential expression signatures aggregated across replicates. So, of course, there are a number of analyses that I have been and will and plan to do as part of the validation of this data set. And I've roughly broken them down into 3 groups. So first, we want to make sure that the data is of good technical quality. So these are just kind of assessing our standard technical quality measurements. Next, I think it's important to assess, is the data of good functional quality? So here I mean, did the knockout actually occur? Are replicates correlated? Are the signatures strong? Is there agreement between guides targeting the same gene? And can we kind of orthogonally validate at least a reasonable proportion of these signatures using our other data? Remember that we chose at least a subset of the genes that we're profiling to overlap with those we already have signatures for in both shRNA and compound space. And then next, and also kind of in parallel, I've been looking at what I'm calling kind of more exploratory questions. So I've been looking into quantifying the effects of the components of the CRISPR screen. So for instance, the derivatization process, we know that Cas9 is a pretty big protein. And so we've kind of been looking into what effect that has on expression space, characterizing the effect of the dual virus treatments and so on the ultimate signatures that we get when we treat with the targeting guides. I've also begun looking at the presence of the copy number dependent cutting effect that some of our colleagues reported in the Achilles data, I think several months ago now. And then I'll be looking at the performance of single guide signatures versus consensus sgRNA signatures. So these would be analogous to the consensus shRNA signatures that I told you all about earlier. I'll be looking at loss of function signatures of genes in the same pathways. If we can particularly see commonalities and differences between them, this could, suggest some really exciting isogenic setups for future experiments. I'll be comparing with external data sets. We're very lucky to have very complementary viability data from Achilles that I will definitely be comparing our data to. And, last but perhaps most excitingly, I'll be combining this data set with the shRNA and compound data we already have to nominate novel and hopefully first-in-class therapeutic hypotheses for people to explore further. And we will be making this data available so other people will be able to eventually do the same. But today, I'll be focusing on kind of points 1 and 2. So is the cohort we've generated so far of good technical quality, and how is it doing from a functional quality standpoint? So to contextualize this for you a little bit, the data that I'll be talking about today represents 8 virus plates treated in 8 cell lines. Unfortunately, when I aggregated this data, we hadn't completed data generation of all these virus plates in all the cell lines. So you can see that the counts in different cell lines are slightly unequal. But overall, this represents 1,781 unique genes. And they were, for the most part, profiled at about 2 guides per gene. So these plate maps represent kind of like a heat map view of the virus plates that we received from our genetic perturbations platform. And these represent virus titer levels. You can see they're pretty high overall. You can kind of disregard the numbers at right. I basically colored these by, if they're gray or if they're blue, that means titer is low. And if they're orange or red, that means titer is at an acceptable level. And you can see pretty clearly that although there are some exceptions, for the most part, the virus titer was good. We also, as I mentioned, measure infection efficiency by comparing Puro versus no Puro plates. And here at left, I have an example of one. This is our XPR8 virus plate treated in A375. For context, at right, I have just the titer plate, and you can see that infection efficiency is pretty good overall. So we generally use a value of 0.7 as our cutoff. Again, that's what I set to be the gray value. So if a well is, red or reddish, that means it has an acceptably high infection efficiency level, and gray or blue means it's a little low. Just in aggregate, these are the infection efficiency ratios overall. You can see that, looking to the right of the dotted line, which represents infection efficiency of 0.7, it's quite high overall, even if you separate by at left virus plate or at right cell lines. So that's great. We have a number of technical QC measures that we also check. I'm not going to bore you all with the details, but again, we're happy to discuss it more in depth. But just to show you a couple of them, the bead counts, we use Luminex beads to measure expression. We're very high in this data, which is great. Obviously, you can't measure expression if you don't have anything to measure expression with. So we generally filter out any wells that have bead counts of less than 20. And as you can see in the distribution plot, there weren't very many of them to begin with, but none of those were included moving forward anyway. We also have a couple gene sets that we use for standard QC. They're called invariant gene sets. And all these are sets of genes that we found to generally have a very consistent expression level. So here I've shown you the invariant gene set 10. So these are genes that we expect to have pretty high expression regardless of the cellular context. So we use them as kind of like a benchmark just to make sure that the expression magnitude is at reasonable levels. And again, you can see that for the most part, by this QC measure, the data looks good. So that's great. One thing that I'm still exploring is we have seen that when you look at within plate reproducibility, the values tend to be lower than what we've previously seen. So on the one hand, this kind of makes sense because first of all, we aren't getting perfect infection efficiency, and unfortunately, even having cells be infected does not necessarily imply that the knockout is actually occurring. But it's something that I'm still working to explain computationally, and if anyone has any thoughts or prior experiences looking at this type of data and reproducibility in it, I would love to hear any ideas in terms of where to look as well. Or not. Moving on, I'm going to be talking about kind of more the individual signatures. But first, I kind of wanted to introduce a metric that we use very commonly. It's called TAS. It stands for Transcriptional Activity Score. And all this is a composite of signature strength and reproducibility. So the exact formula is down here at right. You can see that pretty much it's just a combination of signature strength, that's all SSN gene is, and the replicate reproducibility is what CCQ75 means, scaled by the number of replicates included in the signature. So at top, just for reference, we've plotted tasks. So these plots are, the y-axis is signature strength, the x-axis is replicate reproducibility, and the coloring scheme is TAS. So anything that is not gray is a bioactive TAS, and the darker colors means a stronger TAS. And you can kind of just see, for context, the typical TAS values we get for our different reagents. So in the middle are compounds. To the right of it is overexpression signatures, and to the right of that is shRNA signatures. And you can see again, for instance, that the shRNA signatures are really reproducible and strong. A lot of that is because of their off-target effects. And again, just for the future, we generally use the rule of thumb that a TAS of above 0.2 is what we would consider to be bioactive. It pretty much corresponds to the Taylor values for DMSOs. So anything above that is our usual filter for bioactivity. Any questions?
Yeah, I had a question in terms of the CRISPR's targeting genes, right? So there are multiple guides per gene, correct?
Yes.
And is there a cutoff that you're using as to how many guides you would require per gene in order to include this in the analysis?
That's a good question. We always profile in at least 2 guides per gene. In our prior kind of pilot experiments, we actually did more than that, but we found that the guide signatures were actually reasonably concordant, so we didn't need to include as many to get the same data. From a computational standpoint, I'm still in the process of looking into how results change when you basically aggregate guide signatures versus look at them individually. So preliminarily, yes, I will only be looking at ones that have multiple guide signatures to do that analysis. And then based on what I find, we'll kind of proceed accordingly. Does that make sense?
Okay. Yep. Thank you.
No problem. Anything else? All right, back to the data. So this is kind of the same plot. It's for our CRISPR cohort. And at left, you can see our non-targeting guides. That's what control vector means. And you can see that overall, the reproducibility is low, their signature strength is low, and there are some TAS values that are high, but for them, those are definitely in the minority. So that's basically what we would like to see in, like a null. At right is our test data. as I was kind of alluding to when I was showing you the reproducibility plots, it's certainly variable. There are some super strong signatures that are also really reproducible. It's what we'd like to see all our data look like in a perfect world. But there are also some that have really low reproducibility and particularly unusually really low reproducibility and high signature strength, which I found particularly unusual. For instance, choosing guides to query, I pretty much filtered all the guides with replicate reproducibility values of below 0.2 because that's what we generally use as our cutoff for a reproducible signature. And it doesn't really make sense to, reach conclusions about a signature that isn't reproducible because you don't know what it represents. So here I've just plotted the TAS values, which is what the colors were in the previous plots. And kind of similarly to what I was saying before, you can see that the non-targeting guides have an overall, left-shifted TAS distribution, and the targeting guides have a slightly right-shifted one. We're particularly interested in this range, but really I'll be looking at all guides with TAS values of 0.2, and actually I'm also looking at the TAS metric overall because it was developed in the context of our previously existing perturbagens, and it's also entirely possible that, it might not be the most appropriate measure for the CRISPR data, but for now I'm continuing to use it in addition to other measures. So looking at knockout performance, here I've plotted our reference guides in the dark blue. So remember, these are the guides that we validated in our pilot experiments as being well-performing in our assays. And then the light blue lines represent knockout in our test guides. So again, remember that a rank of below 10 is what we generally consider to be good. And if you look at these CDF plots, you can see that some of our best guides get values or, get knockout values of about 80 to 90%. And then overall, the data actually looks really great. We're getting about 70% knockout for the most part when you just look at every well across the cell lines that I had data for. I'm sorry. Also, very important to note, all of these plots are only for our directly measured genes, so our landmarks. So it's a subset of the data. I just wanted to validate knockout performance in the things that we're directly measuring, and I'm also doing the majority of my preliminary analysis in that. And then once we have confidence in our directly measured data, I'll be kind of expanding the analyses to the cohort overall. So yes, of our directly measured genes, about 70% of the guides seem to be working, which is great. We're pretty excited about it. And just to contextualize that value a little bit, this is the same plot, but it's the best guide of all the guides we had targeting a gene. And if you kind of flip between them, you see that the values change a little bit, but not a lot. And to me, this is particularly promising because it indicates, not that there's just like a subset of guides that are, for some reason, way better, but the guides overall, at least from a knockout standpoint, are working quite well. So here I looked at knockout performance across cell lines. We haven't fully finished profiling in all 10 yet, but we will be profiling these in all 10 of the cell lines I introduced earlier. And this bar plot here just shows the number of cell lines that had at least one well-performing guide for each of the landmark genes that were profiled in it. Let me know if that doesn't make any sense. But the main point of it is that for the most part, guides didn't just work in one cell line. They worked in, not all of them for the most part, but a subset of them. And if you think about how different those cellular contexts are, that makes a lot of sense, right? Like baseline expression of these genes is going to be pretty different across these cellular contexts. And particularly if a cell line happens to have low baseline expression in a gene, we don't really expect to get a strong knockout signature out of that because the knockout of the low baseline expression gene is pretty similar to the, standard cell state. But we're also pretty excited about this because it indicates that, there is value in profiling across all these different cell lines. So as kind of like a last validation for this cohort, I queried them against the signatures that we already had profiled in the same genes. So first, we kind of want to make sure that TAS, so our signature strength metric, tracks with connectivity scores. So these two plots show you that the connectivity scores for expected connections, so for shRNA, sgRNA, for shRNA and compound signatures that are targeting the same gene as the sgRNA signature that I queried, the connectivity score is slightly higher than what we would expect overall. The overall distribution is the one at right, and the one at left is kind of the connectivity scores of the expected connections only. Does that make sense? Great. So worth noting is that, this is a subset of the data again, but preliminarily the results of these are promising. So here are some examples of genes that I found that connected both to their shRNA signature and to compound signatures of small molecules that we've annotated as targeting the same gene. So for instance, the MDM2 loss of function signature connected to an MDM2 inhibitor, which is exactly what we expect and what we would like to see to kind of have faith in our data. Because the data that we're querying against in this context was made, at an entirely separate time. It's totally different reagents. And it just, adds validation that the signatures we're getting are actually what we expect them to be. So I think that's very promising. And I am looking forward to examining the data further. I also really wanted to thank the team that I work with. I'm presenting on all their behalfs. Mukta, Desiree, and Dan have done a lot of great work generating our data. Ian did a lot of preliminary work with our shRNA data, especially. And then John, Federica, Dave, and Paquita work in other groups at the Broad and pick apart my analyses and give me suggestions on a fairly frequent basis. And of course, thank you to the CMAP team overall for all of their ideas and support as well. That's all I have. Any questions?
So this is Ajay. I'm curious whether any of your genes overlap with, say, the COMP project, which does similar things in mice and look for phenotypic effects. Did you do any cross comparisons like that?
I haven't yet compared to other projects. As I said, the data is definitely still in the generation process, but I'm happy to look into that.
And I think you went through it and I might have missed it. But if you look at sort of the core pathways, the signaling pathways across cell lines, how reproducible is it between different cell lines that you're seeing?
How reproducible are core signaling pathways between cell lines? Is that your question?
Yeah, with your guide perturbations. I mean, are they pretty consistent between cell lines, or do you think like a lot of variability between them?
I haven't really looked into that with this data yet. We have done prior experiments in MCF10A using isogenic lines that we just bought from Horizon, and we were able to find some pathways being represented, but that was a pretty small data set. So I guess stay tuned is my answer.
Yeah, just to add to that, we do have compound data for compounds that target a number of the core biological pathways. And in general, we do see that they give similar signatures across cell lines. Obviously, that's a different perturbation type. So I think it'll be interesting to see how that compares with something like CRISPR, where you might be perturbing the same pathway, but in a very different way. So we don't have an answer for that yet, as Juana said, but that'll be something that we'll look into going forward.
Thanks.
Anything else? Great.
All right. So one of the things I am supposed to remember to do is that I don't have that information. Does anyone know who is up for next month presentation? Or if somebody can look
OHS. Hi, Ajay. OHSU is up for next month.
All right, great. So you know that. That means
Yep.
I don't have to remind you again. Okay, thank you. So this next agenda item I had was to see if there are any updates on any new data that has been generated for the MCF10A project.
From us or in general?
In general, everyone.
From our end, I'm not aware of any updates. I do know we tried derivatizing MCF10A. We were originally thinking about including it in this CRISPR screen, actually, and the cells were really unhappy once they were derivatized, so we did not continue with that going forward.
Wonder, did you guys use a lentivirus to try to do that? You mean like making them stably express Cas9?
Yep. Yes, we did.
Yeah, so I mean, this is Mark from Sunlight. Every time I've tried to make a stable cell line with 10A without a lentivirus, like by transfection or some other means, they, or flow sorting even, I tend to get that same result where they just don't like it at all. So that's why I was curious if you use lentivirus or not.
Yeah, we did. It's good to know that
Okay.
it's just us.
This is Jim from OHSU. We've now run MCF10A on the microenvironment microarrays in the presence of trametinib, and we have one potential microenvironment condition that looks like it may confer resistance to trametinib, but we're in the process of validating that. So it's very preliminary, but maybe there's something there.
Oh, actually, I have another one. I'm not sure if this falls under the category of the MCF10A core project, although it does involve MCF10A. I haven't participated in these meetings in the past, but we did do a follow-up. We just finished data generation for a follow-up experiment to an MCF10A pilot that we did about 1.5 years ago now. Where we have isogenic knockouts in MCF10A lines, and we're going to see if the isogenic lines recover expected connections from compounds that we think will have differential expression activity in one or more of the lines specifically. And I also, before data generation, did an analysis where I computationally nominated some compounds that I expect would have differential effects in isogenic contexts, and so I'll be validating how well that worked. Basically to see if an isogenic setup is viable for experiments in our assay.
Okay. If there's nothing else, we all have 15 extra minutes to add it back to our life. Thank you all. Talk to you next time.
Thanks.
All right. Bye.
Thanks, Ajay. Bye.
Thank you.